Hi all, we are Group 4 and we would like to pitch our company to Waste Wizards, which addresses removal of plastic pollutants and its byproducts. Our company's vision is to free nature and human from forever chemicals, and our mission is to provide Australian industries with a scalable, on-site bioremediation solution that detoxifies waste. Thus, our title, a yeast-based solution to PFAS pollution. Let's talk about this persuasive threat known as per-polyfluoroalkyl substance or PFAS. These are a group of synthetic chemicals used since the 1940s in the countless products for their water and heat resistant properties. One of its key types is PFOA, perfluorooctanoic acid. The history of PFAS began with innovation but quickly evolved into a global environmental crisis. They aptly named them forever chemicals because of their strong CF bonds that are hard to break down in the environment as well as in the body. They accumulate over time. Exposure is linked to serious health issues, increased risk of testicular and kidney cancer, and impacts on liver function and cholesterol. Environmentally, they contaminate our soil, water, and wildlife, persisting for thousands of years. Let me give you a recent example. Just last year, it was revealed that residents in the Blue Mountains, which is a popular tourist destination in Sydney, has been drinking contaminated water with PFAS for decades, and this water has been serving for some 30,000 people. Blood tests of residents reveal that PFAS levels nearly was 50 times more than what's considered safe in their blood. So this isn't just a distant problem, it's on our doorstep, impacting real life communities and highlighting the urgent need for action.
Thank you, Rudrani. So the current solution to the problem of PFAS is like there are many technologies actually involved for the degradation. So first one is incineration, landfilling, absorption or manufacture or use of granular carbon, active carbon inclination. So the main pollutants which are involved are PFAS byproducts such as landfill gas, leachates, or like incomplete decomposition, or like any like sludge disposal, etc. So there are many apart from being advantages, there are also some disadvantages associated with these technologies. The main disadvantage of the incarnation is that it has a danger of recontaminating of all natural resources such as soil, water, and air. And coming to the landfilling, we have secondary waste streams which are which have the risk of respreading the entire PFAS across an area. And coming to the absorption, the spent carbon actually requires a safe disposal if left untreated for a larger period of time. So for the problem, we have actually addressed a solution for it. So we prepared an engineered yeast that actually secretes dehydrogenase 1 enzyme that is originated from Delphia acidovirens to break down the PFAS using forever chemicals as proposed by Harris in his paper in 2022. So our system usually enhanced with the support modules for electron supply and cellular detoxification, thus creating a robust and self-sustaining biocatalyst for on-site industrial waste treatment. So our solution mainly focuses on the critical environmental challenges for per and polyfluorinated alkyl substances by employing a synthetically engineered strain of Saccharomyces cerevisiae as a self-sustaining whole biocatalyst. So the leading chemical companies that usually degrade the PFA in Australia and around the world are as follows: ScienceDev, Epoch Enviro, and Adventia. ScienceDev is an Australian company that is used to treatment for contamination of PFAS, while Epic Enviro is also an Australian company which is expertised in the remediating of PFAS, which has its manufacturing basis in the US. And Venture is well expertised that heavily relies on developing technologies for the bioremediation of soils that were contaminated with PFAS.
Talking about genetic design strategy for engineering yeast to degrade PFAS pollutants, we used we started with a high copy plasmid PRS426, which allows for strong gene expression and episomal maintenance in the yeast. This backbone gives us high protein yield without integrating into the genome. To build our genetic construct, we used benchling. It made it easier to manage multiple transcriptional units. For assembly, we used modular Gibson assembly. It's ideal for synthetic biology because it enables rapid design iterations and flexible part swapping. Now let me walk you through the genetic components we selected. GAL promoters are inducible elements activated by galactose, allowing precise control over gene expression. The alpha factor signal peptide ensures secretion of the enzyme into the environment so it can act outside of the yeast cell. Our key enzyme is DHA type 1, which catalyzes the breakdown of PFAS by cleaving the stable carbon fluoride bonds, enabling bioremediation. We use ADH1 terminators to ensure proper transcriptional termination and TF1 TDH3 promoters for uh, strong constitutive expression. The PDR5 transporters helps the yeast survive in toxic environments by exporting harmful compounds. Finally, CPRs or NCP1 support cytochrome P450 function by transferring electrons from NADPH, which is essential for the enzymatic oxidation.
So coming to the plasmid design, the plasmid was assembled in a PRS 406 with a URA marker. As we can see here, there are three cassettes. The first cassette over the top right, as we can see, is a cassette with DHA1 coupled with alpha signaling peptide. Next is NCP1, which enables NADPH release to facilitate electrons for the process to take action. These two cassettes are induced by GAL promoters, so the system is only functional with presence of galactose. And the PDR5 is also available in cassette 3, which will make sure that toxins are pumped out with an efflux pump. So assembling the whole genome, we used Gibson assembly in Benchling. And these are the list of forward and reverse primers for each cassettes that we use. Going on to the further modifications that we hypothesized but didn't go with were adding genes that make the plasmid more integrated in the yeast and give it more resistance to the waste. For example, we try to add CTT1, which will neutralize the free oxygen H2O2 molecules in the yeast, heat shock proteins, and CUP1 to help with endurance and survival in heavy metal. Then we made Sagna sequencing primers to verify the sequence. These are the forward and reverse, the first and last primers, and in between there will be about 20 to 30 more primers, depending on the size. Then alternative approaches that we could have tried, but we did not, either due to our limitations or due to the limitations of our approach. It was yeast-mediated assembly, which transcribes the gene directly into the chromosome. We could have used Golden Gate assembly as well, which would be more modular, and we could have also gone ahead with Illumina sequencing for the whole sequence of assembled plasmid.
So when the virtual plasmid digest will be validated and then it will be synthesized and ordered for lab use. The plasmid will then be first transformed into E. coli using heat shock method for amplification. And then the transformed colonies will be again plated in a albe agar plates containing ampicillin. And then the positive colonies will be conformed using colony PCR. Next, the plasmid will be introduced into Saccharomyces cerevisiae strain BY4741 using the lithium acetate or PEZ transformation method. The transformed cells will be placed on SDURA media, allowing only successful transformed colonies to grow. This will also be screened using colony PCR to confirm plasmid presence. To confirm enzyme activity of fluoride release assay using colorimetric detection by sigma enriched fluoride colorimetric assay kit will be used. After all the laboratory validation, we will then move into large scale production where it will go through four workflows to be employed. The first one is pre-treatment, where the industrial waste will be filtered and the pH will be adjusted to create a yeast compatible environment. And then the conditioned waste will be added to the bioreactor containing the engineered yeast, where we will be using fed batch fermentation. Upon galactose induction, the DEHA1 enzyme will be expressed and begin degrading PFAOA. And then all of these will be transformed into a second tank where a fluoride ion released will be treated with calcium hydroxide, forming solid insoluble calcium fluoride. Then the final output will consist of a clean, detoxified water and a stable, non-hazardous solid waste, which will again be safely disposed.
Our industrial and commercial strategy is designed for rapid adoption and long-term scalability. We begin by targeting strategic partnerships with companies in sectors like food packaging and plastic manufacturing, which are the key contributors to PFOA pollution. These early adopters will benefit from a turnkey bioremediation system installed directly at their facilities, along with full staff training. To protect our innovation, we will secure intellectual property rights for our engineered yeast strains and plasmids. At the same time, we will accelerate development by partnering with universities through sponsored research agreements, focusing on improving enzyme performance and strain durability. Within the next 5 years, we aim to establish our yeast bioremediation platform as the standard for PFOA treatment in Australia, supported by multiple industrial installations. By year 10, we plan to expand into other PFAS heavy industries such as textiles and firefighting foam and achieve measurable reductions in PFAS levels in key watersheds. For our financial and research timeline, a 12-month on-site pilot trial estimated at approximately 250,000 to 350,000 AUD for equipment, consumables, and personnel. The challenges include adapting to diverse waste types, ensuring long-term plasmid stability, and navigating GMO regulatory approvals. Our response is to develop a stable chromosomally integrated strains and to work closely with the Office of the Gene Technology Regulator while keeping the bioreactor fully contained for safety. Thank you for listening to us.